Thank you very much, for opportunity to present our work. And yeah, so I'll start the very short introduction, show why we are so desperate about why we really want to be good at modeling regulatory regions, and it is about predicting the effects of clinically significant variants in um, human genome. And actually, as we see, the main part of clinically significant variants in human gene, they occur in regulatory regions, not coding regions. And while the main effort for now is concentrated on predicting the effects of coding regions, it will be really fascinating to be able to predict the effect of non-coding ones. And actually, for now, usually people use sector activity model approach to do that. And this approach is two-step approach. Like firstly, you train a model to predict multiple epigenomic tracks using sequence only. And after that, after you have this model trained, you use it in zero-shot manner or using some fine-tuning to predict the effect of regulatory variance on gene expression. But unfortunately, for now, we do know that there are many problems with that approach. First of all, it failed to account for mutations in distant regions. It has really severe problems in accounting for individual genome variation, and it can predict cell-specific regulatory regions reliably. Also, if one train even the same model architecture but using different random seeds, such true models, they tend to disagree on the effect of many regulatory variants, and sometimes they do predict pretty opposite effects. So, one of the explanation for that problem and the ways to suggesting the way to solve it is like that the genome is just too small to account for all possible combinations, orientations, and spacing of approximately 150, 500 transcriptional factor sites in human genome. And so we just need more information than is contained in genome. And actually, there is a way to measure the activity for thousands, tens of thousands, and millions of random or designed sequences at once, and it is massively parallel reporter assays. But for now, there is no commonly accepted way to process that data, to train model using that data. And so the objective of I already mentioned Dream 2022 competition was to come up with such a method. And organizers provided us with 6 million 80 nucleotide sequences with measured effect on expression in yeast cells using flow cytometry. And for each sequence during the experiment, organizers they find the beans in which this sequence occur after sequencing, and they presented us with the average expression for each of such sequence, average bean for each of such sequence. And also to make maybe competition more fair, organizers they restricted us from using any ensembles or pre-trained models, so no sector activity models in this competition. And to solve the problem of predicting the effect of such small regulatory elements on the activity, we decided to use architectures from computer vision. And we tried a few architectures, and we finally found out that EfficientNet v2 was like the best choice. And actually, in computer vision, at the time of competition, it was the SOTA solution. in terms both of performance and training, and also it was really small, and so we can just care less about overfitting. And actually, even now, yeah, although there are transformers which like behave better maybe in computer vision efficient net v2, according to my friends from industry, it's just a default choice. And so, yeah. And we adapted this architecture to the problem of working with nucleotide sequences. Actually, we've done really small modifications, like for example, we replaced initial residual addition block, usual for such type of architectures, with less frequently used concatenation block. And again, this concatenation block allows us to use a smaller number of weights to work with nucleotide sequences. But actually, the architecture is not the main thing you should care then you are working with such task. It is well known in general machine learning that training methods and scaling strategies matter. You can have like something like very sophisticated architecture, like optimal architecture. But if you use an optimal training method, you likely result in suboptimal results and just bad performance, maybe below average. And so after just we tested multiple training schedules, and the result was that one cycle learning rate coupled with RW optimizer proposed by FastAI training course, it was just the best choice. And it greatly increased actually the performance of our solution. And what was also important that actually in natural language processing, they use the same learning rate schedule. They name it slightly differently, like they have warm-up steps plus like assign and annealing, but actually it's like the same. Like so, one cycle learning rate is just the default choice in general machine learning at all. And what was also very important is that we know, according to Demis Hassabis, that for an important problem in biology, you should throw all your knowledge about all your domain knowledge, and you should care about your problem, not about how general your solution is. And so we tried to make our solution informed about biological side of problem as much as we can. And first of all, we decided to modify the way we provide our sequence to the model. So additionally to one hot encoding, we provided two channels. First channel informed our model whether sequence come from some noisier part of the data or from the more clear one. It's just a really good solution sometimes just to allow your model to disentangle this noisier data from the more reliable parts of your data set. And the second part was about adding reverse channel. So in regulatory genomics, it's common to use reverse complement augmentation to enhance your model. But actually, uh, in case then you are working with promoters, we know that many effects of promoter sequences, they are strand specific. Some don't, but many of them they are some not, but some of them they are really strand specific, and so it's a really good idea to allow our model to decide if the effect it trying to predict is strength specific or not for current promoter. Also, it was really interesting to find out that according to the research done by organizers before the challenge, actually the expression they provided us with uh, is distributed normally. And we can try to replace point estimate of the expression with some like distribution, normal distribution with two parameters. The parameters like standard deviation, we have chosen it using some like just the cross validation, and the mean of this normal distribution, we decided uh, to, to set it equal to expression provided by the organizers plus 0.5, and it's like some analog of continuity correction. And once we have, for each sequence, we have such distribution of the expression, we can calculate the probability of sequence falling at each being in the experiment. And after that, during training, we can ask our model to predict not single value, like single expression, but to predict the probabilities of our sequence falling in these bins. And during evaluation stage, we can use these probabilities to predict single expression value using soft argmax approach. And yeah, you just actually multiply the outputs of softmax function, so the probabilities of a sequence falling in each bin by the number of this bin, and thus you obtain like expression value. Okay, so our solution, yes, secured the first place in the competition. And what was more important, that our solution was really stable, and it was not like our solution was best only overall, but it was best in each single subcategory in the challenge. And especially it was really fascinating to observe that it is really good in predicting the effects of single nucleotide variants. Next, we tried to check if we can apply our solution to other data sets of the same type. And fortunately, yes, again, organizers of the challenge, they had multiple papers with different data sets. On yeast, about activity of regulatory regions in yeast. And so we again we retrained our model on these data sets, and we observed that again it outperformed existing solutions, and it is really good again in predicting the effects of single nucleotide variants. After that, with organizers, we tried to optimize our solutions. Like the idea was that, okay, we have solutions from multiple teams, and so we can try to split it in different blocks and combine these blocks in different ways, and like the result might be better than any single model. And it turns out that actually it's really hard to beat LegNet, even using like ideas from other solutions. And yeah, actually on original data set, it's mostly impossible. But, and it's really important, if we use training scheme and problem reformulation as we suggested for LegNet architecture, uh, for other architectures suggested by second and first places, it greatly improves the quality of the solution. Like it's really hard to distinguish it now from the quality of the original solution, our solution. But at the same time, I should mention that actually our solution like uses at least two times less parameters than other solutions of the other participants, and so it's really parameter efficient, and we again can be less we can care less about some problems with overfitting and so on. And what is also important, it is really fast to train and to perform any predictions using this solution. So why not to try now to ensemble it? So let's just train different versions of our model using just different random seeds. And as we see, actually adding additional models to like averaging our model different predictions of different versions of our model result in slight yet significant increase in model performance. So again, as our model is really fast to train and really fast to predict, there is no problem just to for any challenge just to train multiple versions of this model and just to use ensemble. Okay, so but it was about yeasts. Like yeasts a really good organism, but we actually really want to work with human data. And yeah, actually, Agwal et al. They published data on the effect of expression of many human regulatory sequences. And so it was like a challenge for us to test if our model could do better than models I already presented in this paper. And again, it was the like the main part of our solution here was about like uh, thinking about the problem. And the main part was that actually they measured activity of longer sequences. And so we should increase the receptive field of our neural network. And at the same time, the amount of more of measured regulatory sequences is lower, like uh, 10 times maybe or more. And so we can just increase the number of parameters in our neural network. So we should somehow increase the receptive field in the different way. We tried different approaches, but finally we found out that just adding max pooling layers helps a lot. And also, and this is also very important actually, that uh, our solution based of like classification and so on, soft classification, reformulation, it was about experiment. And here there is no bins, there is no classification. So we just substituted our classification head with simple regression one. And the results were pretty amazing that, yeah, actually we in 2 out of 3 data sets, we are able to surpass the quality of models trained on many epigenetic tracks like Sei and Enformer. And on the third data set, we are really competitive. We provide a very competitive solution with these models, but unfortunately, we failed to overcome their performance. But actually, this data set is quite noisy and it's really small compared to other 2 data sets. And again, I should mention that actually, in case of Enformer, LegNet just use 200 times fewer parameters than Enformer. Okay, so yeah, it's like this meme. Like we just can use way less parameters to do the same task. Okay, but and what about regulatory grammar learned by LegNet approach? And again, our collaborators, they and we tried to measure this in different tasks, and it seems like LegNet really good at predicting the results of saturation homogeneity data from Kirch et al., and it performs on par with Enformer and other models. When we try to use data from amazing databases, Adastra and Odacha, developed in our lab. And these databases, they contain information about allele-specific events, about DNA's accessibility and transcription factor binding. And it again, we have shown that our model is able to predict allele-specific events. So again, it's really nice. And also, it was interesting if our model can account properly uh, uh, for interactions between different um transcription factor sites. And it seems like, yeah, it correctly learned this aspect of regulatory grammar. Like it is really good in predicting the effect of just placing in one sequence many sites of the same transcription factor. Like we clearly observe saturation effect, and it's like in experimental data, and our model is good at modeling it. And if we talk about interactions between with about transcription fact binding sites of different transcription factors, we again see that our model is good at predicting both like sub log additive interactions. Like then you need only one transcription factor binding site to activate your sequence, and adding additional binding site doesn't do anything. And again, and super log additive before interaction, then adding uh, transcription binding sites of two transcriptional factor result uh, in much higher regulatory activity of the sequence than we expected by log additive model. And again, our model can predict that. Okay, so but is Enformer really useless? Like, is it possible, for example, to combine features from Enformer with LegNet architecture? Like we do know that if we try to apply our model to smaller libraries from the same paper, actually our model performs like sometimes on par with this with sector activity models, but sometimes worse than them. And so it will be really, and it is mostly about like the amount of data. And we do know that for many MPRA experiments, we just don't have so much data as it was in Agwal study. Okay, so let's try to do that. And the answer for that, yes, it's LegFormer, developed by my student. We can just use features from Enformer model and add them to the features extracted by LegNet convolutional part. And after that, we pass them jointly to the sequence of fully connected layers and learn this model train this model to predict regulatory activity of the element. And actually, the results, they're really good, like for large library. We were in cases of HEPG2 and the second cell line, we were really good in predicting the regulatory activity, and we were even better than Informer. But if we add features from Informer, they're even better. So again, so it's really good idea to combine these features. In case of other libraries, there we were inferior to Informer-based solution. We now on par with Informer-based solution, and sometimes we actually can perform even better than Informer. So it's a really good idea sometimes just to use both features from Informer and like a cleverly designed small convolutional neural network. Okay. And actually, then you are talking with machine learning specialists. They really like to cite this work, like about beta lesson of machine learning. Like, and the main idea of this paper is that there is no need to think about your data, the nature of your data, and so on. You just can stack more layers. You can just add more data, and the results, they will be better and better. And at least now, for regulatory genomics, we can state that actually this myth is busted. So there is no such rule and lesson in biology. You should care enough about domain knowledge. Okay. And so now let's move to the second part of my talk, and it will be about generating sequences with desired properties. And it's really important just because it can be used, and actually it is used in biotechnology and ability to generate sequences with cell-specific activity is highly required by personalized medicine. And also, if we have like a really good generative model, we can use it for active learning to further enhance our methods, to narrow this search space of sequences we need to measure in experiment to improve our understanding of regulatory grammar. And the main approach used nowadays is for generation is diffusion models. Like you gradually add noise to your image or to any other object until it just become undistinguishable from random noise. And after that, you learn model to restore like the original image. And it turns out that if properly trained, such model can be used to generate new images just from any random noise. But there is a problem that if we are working with nucleotide sequences, they come from a categorical distribution, and most diffusion models they are working with real values, real value data, and so you just can't add no, and they are working with normal with adding normal Gaussian noise. You can't do that directly for categorical distributions. But there is a paper called Diffusion. Their author shows that actually you can design your own diffusion process, and as long as you can just gradually add any kind of noise to your object, uh, you can use such like procedure as some kind of diffusion process. We can like call it cold diffusion process or like diffusion-like process, but yeah, you can do that. And the generation, and you can train model using such process to generate new objects. And so we decided to use like a very simple diffusion process. Just let's start from sequence containing some relevant information, and we will just gradually add single mutation to the sequence until it becomes completely random. And yeah, there will be some like matching letters in this sequence, nucleotides, but it's just about like there will mismatches they will be just due to chance, and there is clearly no sign of CTCF motif. And the task of our model will be to, to try to restore this original sequence given the fact that it contains CTCF model, or it was active uh, due to binding of CTCF factor. And so we trained a model to restore original sequence using mutated one, and also we provided our model with the expression of the original regulatory element. And yeah, and so we optimized cross entropy between predicted sequences and original one. And once we trained such a model, it was really easy to use it to generate new sequences from random one. We just provide our model with random sequence and desired expression. We just do simple diffusion generating process as was described for even first diffusions, and we get like sequences with desired expression. But is it really works? Actually, work does it really work? Yeah, actually, works, and the correlation between target expression and predicted expression is pretty high. But the problem with that, like test, like with this performance assessment, is that it is done in silico. just because we don't have facilities to generate to measure the activity of these sequences in the lab in experimental settings. So we just can say that, okay, so our model generates sequences which are predicted to be expressed in the way we desire by the other model. It's okay, but there is still room for improvement, I think. And we tried to find people who want to work with us on generating sequences with desired properties. And actually, we do know that regulation and the specificity of sequence cell specificity is controlled at different levels. And yeah, most attempts, most efforts, and it's really rational, are focused on optimizing cell type specificity at promoter level or an enhancer level, like DNA level. But we also can try to optimize cell specificity at transcriptome level by fine-tuning 5' UTRs and 3' UTRs. So our collaborators, they done like a really amazing experiment. They measured the effect of 3' UTRs and 5' UTRs on the expression of our on expression of target protein in many cell lines. And after that, we decided to check first to check could we predict this expression using just LegNet approach. And actually, yes, it is possible. Like we can just train our model to predict activity of our sequence in the current cell line and the difference between this activity in current cell line and the mean activity of this sequence across all cell lines. And if trained this way, our model now, like it changed its name, like now it's Parade. It can be, it is really good in predicting activity of like the effect on expression of 5' UTRs and 3' UTRs. So, okay, we can predict that. So, we can expect at least that our model will be able, like our diffusion-based approach will work. So we adapted it to generation, yeah, again, of the sequences with desired cell specificity. And after that, and yes, we validated it experimentally. And we trained our model on library one. It was like our training data set. And it seems like, and after that, we generated sequences for library two. And actually, library two sequences, they're much, they show like much greater tissue specificity than the original data. So it's really great to observe that and that our approach actually work. Uh, I can like disclose all the details of this study, and the preprint is preparation of on this work, and I hope that we will publish it this month, and you'll be able to read it. And after that, I hope the main author, my good colleague Arseny Zinkevich, will be able to present you with all results of this study, just because it wasn't only about applying diffusion models. It was about applying different algorithms and so on, comparing their performance. And the results, they're really amazing. And yeah, I just, yeah, I hope we will be able to present them soon. I want to thank all people who participated in this project, and I want to thank actually Vikram Agarwal, because after we reported that LegNet is able to predict very well on human regulatory sequences, he invited us to join his paper, and yeah, it was really amazing to work with him, and yeah, thank you again. So here you can see the conclusions of my talk. And also, if anybody interested, you can follow like GitHub with human LegNet, and you are free to apply it to your uh, tasks. Thank you for your attention. I'll happy to answer any of your questions.